Greetings and welcome back. So on our last lecture, we had talked a lot about the monosaccharides under the umbrella of what are called carbohydrates. Monosaccharides, a few of the examples are shown here, are given in a specific type of formation known as a Fischer projection. We had looked at Fischer projections. There were some rules and criteria for that, which we looked at before. We had classified these monosaccharides written in the Fischer projection as based upon the most oxidized carbon on them, as well as the number of carbons on them as well. So this could be an aldotriose, this would be a ketotetrose, and so on. We got into something new as far as isomers. Also, we talked about the concept of stereochemistry. Stereochemistry being these non-superimposable mirror images, but in every other way they are identical. In a molecule that can bear stereochemistry, the carbon from which all bonds emanate are known as chiral carbons, and we were able to determine what is a chiral carbon, what is not a chiral carbon. Put specifically to monosaccharides, there were frequently a number of chiral carbons in monosaccharides, but we had then looked at a very special chiral carbon in a monosaccharide known as the chiral carbon. The chiral carbon is the chiral carbon, for instance, in this molecule right here, which is furthest from the most oxidized carbon in the molecule, the furthest chiral carbon. And from that chiral carbon, the chiral carbon, we were able to see some what are called D and L configuration, in monosaccharides. So for instance, in this molecule here over on the right, here's the chiral carbon, and we have the hydroxyl on that chiral carbon pointing to the right. Thus, it would be a D. The right is D. If over here on this molecule, here's the chiral carbon. If the hydroxyl pointed to the left, it is an L monosaccharide. Only D monosaccharides exist in nature. Now, interestingly, as we alluded to in the beginning, monosaccharides can be shown here as in a Fischer projection, and it's an open chain. If we look at all 6 of our carbons in this case, they all occur in an open chain. But in fact, in solution, the larger monosaccharides, and by that I mean either 5 or 6 carbon monosaccharides, tend to want to spend most of their time in a cyclic form. In fact, greater than 99% of the time, if we have a dissolved monosaccharide, it spends its time in the cyclic form. Now, just as we showed the way to express an open chain monosaccharide in a Fischer projection here, and all Fischer projections are written exactly the same way, always with that most oxidized carbon towards the top, and all other carbons in a sort of a zipper down the middle. It's always the case. Now, with a cyclic form is always given in what is called a Haworth projection, and here clearly what we see here is a cycle. We'll look at more how these cyclize in a moment, but a Haworth projection of a monosaccharide is always shown in the same way as well. We'll be talking a little bit more about that before. It can be helpful to number the carbons. If we look over on the left-hand side now, the Fischer projection, notice we have these carbons numbered 1, 2, 3, 4, 5, and 6. Well, I can show this an open chain form, not just in a Fischer projection, but also perhaps in a more suggestive form, this piece in the middle where, notice my carbons 1, 2, 3, 4, 5, and 6 are written suggestively now. And that means that this the hydroxyl off of this 5 carbon right here is poised, the oxygen on it anyways, is poised to make a bond with the carbonyl carbon here. So that once that bond forms, what ultimately happens is we get a cycle given over in the Haworth, shown as the Haworth projection over on the right here. Let's take a bit of a closer look now at how our cyclized monosaccharide forms. On the molecule over on, or the rendition over on the left, it shows our suggestive way of drawing the open chain monosaccharide, in which what is expressed is where the C5 hydroxyl is, that fifth carbon down. There was a hydroxyl on there, and here it is pointing out suggestively. What happens is the lone electron pair on, or one of the lone electron pairs on that oxygen forms a bond to that carbonyl carbon on that aldehyde, and that would give 5 bonds around carbon. So one of these double bonds to the oxygen have to go up and sit on oxygen. Ultimately that connects with a hydrogen, but let's look at the connection here. If we form a bond right here, the C5O oxygen is right there, the hydroxyl. That is over on the right hand side, this oxygen right here. That's the ring oxygen from the C5 hydroxyl. Now, the carbon of the carbonyl over on the left-hand side becomes this now undrawn line structure type carbon, the 1 carbon right here. It's not drawn in there, but that's exactly what it is. Indeed, on any Haworth projection, that carbonyl carbon becomes the carbon furthest to the right on the Haworth projection. So ultimately what we have here now is that we have the carbon from the carbonyl is given right there. That used to be the carbon from the carbonyl on the aldehyde over on the left. And look at this hydroxyl right here. The oxygen from the carbonyl is now a hydroxyl. If we look at that, this hydroxyl right here, in this case it's pointing downward in this Haworth projection, but it becomes instructive to look at this connectivity from the standpoint of something that we have looked at before. If we could look at what kind of connectivity we have. If we follow my trace here on this right-hand molecule, this Haworth projection of a monosaccharide, notice I'm going to trace out COCOH. If you remember, that was a hemiacetal connectivity, was COCOH. So now in nature, we see a COCOH hemiacetal linkage. So I'm going to draw that on the molecule on the right up here. So follow me. Carbon, oxygen, carbon, oxygen, hydrogen. COCOH. And indeed, that is a hemiacetal linkage found in nature, in this Haworth projection. Most important on here is going to be the fact that, let me tidy this up a bit here, over on the, we'll go from the left to the right, is going to be this, let's look over at the molecule on the left. This C5 hydroxyl is going to form a bond with this carbonyl, in this case of the aldehyde. That's where we get that. Look over on the right now. That's the bond that is formed. Now we have a cyclic structure. That cyclic structure forces our oxygen from the carbonyl on the left to be an oxygen in a hydroxyl on the right, in this case pointed down. The carbon, the 1 carbon, see if I can, this 1 carbon over here on the right, and the oxygen, which were from the carbonyl on the left, have special names associated with them. They are, the carbon from the carbonyl is given as what is called the anomeric carbon. And the oxygen that used to be from the carbonyl, which is now a hydroxyl, is known as the anomeric oxygen. Anomeric carbon and anomeric oxygen. Always on a Haworth projection, the anomeric carbon and or carbon rather will be the furthest on to the right. Always be the furthest to the right. That ring oxygen on a Haworth projection in a 6-membered ring will always be sort of in the upper right-hand corner. notice that what I call in a Haworth projection is in the 6-carbon monosaccharide, the flag, the C6 flag. There's always going to be a flag sticking up there on these Haworth projections. Yes, it's CH2OH. I call it a flag because it's kind of up there flapping in the breeze. But always, at least in a 6-membered ring on a Haworth projection, all of these things are going to be maintained consistently. Let's take a closer look now at what this cyclization process is all about. And I would bring your attention to the molecule in the middle, this open chain form right here. And because we're in the open chain form, notice that we have free rotation around all of these carbon-carbon single bonds, notably the one with that little red arrow next to it that shows that bond can enjoy free rotation. Now, that is indeed happening always in any molecule of any carbon-carbon single bond, but the ability now of that carbon 1 to carbon 2 bond to freely rotate allows for the aldehyde oxygen there to spin around and around and around while that monosaccharide is open. Once we get to a point where the C5 hydroxyl is going to form the bond with the carbonyl that we see right there, that either locks this aldehyde oxygen up or down and keeps it there. Remember, when we're in a cyclic form, substituents are locked in place. When we're in open chain form, substituents are free to spin around because of the nature of that carbon-carbon single bond. Because of this, what we get now when we look over to the possible structures on the right-hand side is that if you look at the structure upwards on top, that bond was just formed right there, giving our anomeric carbon and anomeric oxygen that we see there, that used to be the carbon and oxygen of the carbonyl. Notice the disposition of this anomeric oxygen. It is pointed down. That would mean that if we look in the central structure again, as this aldehyde oxygen was swinging around, it was caught when that bond formed on the low end, caught facing down, so that anomeric oxygen now on the structure on the upper right is forced down, is caught or captured in a downward formation. Now, similarly, if we had our aldehyde again spinning around, and at the point that the bond was formed between that C5 hydroxyl and that carbonyl carbon, if that aldehyde oxygen was upwards when the bond was formed, that now if we look on the lower right hand side, that anomeric oxygen now would be forced upwards. So we have the possibility of our anomeric oxygens being up or down. Up or down. Now this is the only center which we have a choice at. We can whether our anomeric oxygen is up or down. All of our other hydroxyls and substituents on there, as we go around this ring, this hydroxyl here on the upper right-hand side, this hydroxyl, this and this C6 flag, they're all locked. They never change. The only thing that can be a choice is that anomeric center, that anomeric oxygen, whether down or up when we get a cyclization. Now, we have a way now of identifying. Remember when we looked back, we were able to give a designation for monosaccharides when in the Fisher projection. For instance, over here on the right, I could look for the chiral carbon, and we see that hydroxyl is pointing to the right. That means it is a D designation. If it was pointing to the left, it would be an L designation. So very similar to that when we look over at the two structures on on top of each other over on the right-hand side, we can designate them as either what is considered an alpha anomer if that anomeric oxygen is down, or a beta anomer if that anomeric oxygen in the form of a hydroxyl is upwards. So any of these we can point at now, given we talk about an alpha or a beta anomer. It all depends on looking at that carbon furthest over on the right, in which is our anomeric carbon, and the disposition. When I say disposition, I mean which way is it pointing, upwards or downwards? If it's pointing upwards, it is a beta anomer. If it's pointing downwards, it's an alpha anomer. Now, we call these the anomeric carbons and anomeric oxygens. What we have here are anomers in general. This is an anomer. This is also an anomer. Now, why are we using that term? Let me tidy this slide up a little bit here, and we'll start talking about that. Notice, going from, let me fix this up here. In the central structure we have here in this open chain form, again, here's the open chain form, we had a situation where our bond was going to form and our aldehyde was spinning around and around and around. And it can, when we get closure of that ring, we can have either our alpha anomer formed or our beta anomer formed. Now, let's think about this for a moment. When we have our open chain form, we have our aldehyde spinning around. When we have our closed form, we have either anomeric oxygen down or anomeric oxygen up, giving the alpha or beta respectively. Notice the arrows going from that central open chain form over to the cyclic forms. These arrows here, and let me highlight them, they are back and forth, back and forth. So what we have here is we have an equilibrium between the open chain form and the closed chain form, whether they be the alpha anomer or the beta anomer. Now, so that essentially says that our two anomers can interconvert between one another. We can interconvert between one another, because if we were stuck in the, let's see, let me just clean this up. If we happen to be in the beta anomer, well, this beta anomer can open up again. It is labile. That hemiacetal linkage within this is labile, means it can open up and reform its aldehyde and the hydroxyl there that originally formed that bond. So now we have an open chain form again, which immediately starts that newly formed aldehyde spinning around and around, so that we could perhaps then close it off, but in this case the oxygen would be down and would be stuck in the alpha form. So these alpha and beta anomers, again, let me tidy this up a little bit. These alpha and beta anomers themselves can interconvert. They can interconvert. Why? Because both of them are constantly opening back up and closing, opening back up and closing, always through this single bonded intermediate, which immediately gets that aldehyde oxygen flapping around, around and around and around, so that we can lock into either the alpha form, the alpha anomer, or the beta anomer. Now, locking these anomers from an open chain form into a ring brings back is somewhat familiar to the cyclic structures we saw before in geometric isomers, when we were talking about geometric isomers that we had with cyclic alkanes in particular, cannot interconvert from cis to trans. Now, if you remember, if we look at this as just a representation, I've cleaned up the general structure of a Haworth projection. This is not a monosaccharide, but just to give give a comparison, we have a 6-membered ring, an alkane ring, cycloalkane, with 2 substituents on it, 2 hydroxyl substituents, just for the examples. Now this cis isomer has our 2 hydroxyls on the same side of the ring, whereas over here on the right, the trans isomer has the hydroxyls one on one side, one on the other. We're sort of used to that from organic chemistry. Importantly, now we cannot interconvert between the two. They were 2 completely different compounds. They cannot interconvert. Geometric isomers cannot interconvert. However, these anomers are familiar. They're similar to geometric isomers. So I am claiming that anomers can be thought of, in tongue-in-cheek, can be thought of like leaky geometric isomers. Leaky means one can leak into the other. If it were in the case of geometric isomers, the cis isomer could perhaps leak into the trans isomer, which we know is not true with geometric isomers. Now, this is, again, a cleaned up slide, sort of a Haworth-like projection, just to show you the difference here, in that if we look at the structure over here on the left, our alpha anomer. Why would that be considered an alpha anomer? Well, look at this anomeric carbon over on the right, and this anomeric oxygen is disposed down. Therefore, it is considered an alpha anomer. So we have an alpha anomer. Now, remember, anomers can reform. Let's see if I can. So we have these curved arrows, so we can reform this aldehyde and break this bond that we had here to give our central central structure right here, in which once we do that, then we can have our aldehyde here continue around and around and around. So we're spinning now, again, until such a time as we collapse this bond again and form the cyclic structure, and then perhaps we could catch that aldehyde oxygen on the upswing here, and then over on the right-hand side, look at that, we caught our oxygen up top here now, so that would be the beta anomer. Now, the only reason I'm saying this is because we have situations where we have cycles. This is a cycle. This is also a cycle. And again, let me, my apologies, I'll try and clean up this, some of this stuff here. We have our cycles, but these cycles can open up. If the cycles can open up, that means we can reform our aldehyde. Again, and once that aldehyde is reformed, it starts spinning around and around and around again until such a time as that hydroxyl closes on that then carbonyl carbon, and we form either the alpha or the beta anomer again. The only reason I'm comparing these to geometric isomers is because we have cycles here, but they're labile cycles because we have this oxygen in the ring. It's a labile cycle because we have this oxygen in the ring. That does not happen with geometric isomers. We either have the trans or the cis, and that is it. We do not interconvert between the two. Wow, look at this. We've totally made a mess here of this entire slide. Let me try and erase a little bit so things can be a little better. All right, on to the next thing. Just to get some practice with being able to label anomers in cyclic monosaccharides, here we have 3 representative ones. We got a couple that we're a little bit familiar with. This is sort of 5-membered rings, right? Or I'm sorry, 6-membered rings. Here's a 6-membered ring. Look at this one over here on the right. This is a 5-membered ring Haworth projection. There is a mechanism for that as well. It looks different than a 6-membered ring Haworth projection. We'll talk about that in a while. But what we're doing right now really is to just determine on each of these what type of anomer is being shown. And to do that, we always look to the carbon, and it will always be there on a Haworth projection, the carbon furthest to the right. Over here at the molecule on the left, there's the carbon furthest on the right. Hydroxyl pointed downwards, thus it is an alpha anomer. Again, in the center structure, the farthest carbon to the right, that's the anomeric carbon. The anomeric oxygen is disposed upwards. Therefore, that is a beta anomer. On this one on the right, a very strange one, our 5-membered ring, we can still find that ring carbon that's furthest over to the right. There it is. And that OH, that hydroxyl, is disposed upwards. Thus, this would be a beta anomer as well. Let's loop a couple of things in here. The first one is that here on this structure that we're showing on the left-hand side, we have our standard Fischer projection in which we show that it can indeed be cyclized into two different anomers. One when we have the alpha anomer and one in which we have the beta anomer. The alpha anomer has hydroxyl disposed down, beta anomer hydroxyl disposed upwards. Now, importantly, if we look at we got some names here, so we're going to start to get familiar with some names of monosaccharides. This is the structure we see here, various forms of glucose. Glucose is the major energy source in animal systems. We'll be talking about that a little more in further chapters, but notice in each case, it shows it is D-glucose. From here on out, we're always going to be doing D-glucose to the point where I don't even need to call it D-glucose, because if you remember, the D designation of glucose or any monosaccharide are the only ones that occur in nature. Because this is biochemistry, these are natural processes, we only care about what is happening in nature. So although you may see these D designations in here, I'll frequently just omit them and just call it glucose rather than D-glucose, because it's always going to be D. So with that out of the way, here we see we can come into either an alpha designation or alpha anomer or a beta anomer, which we looked at before. We are going to be called upon to recognize up to 5 representative monosaccharides, one of which is glucose. I will only ever ask you to recognize, in this case, glucose when in the cyclic form, whether it's alpha or beta. I will never have you recognize glucose in the Fischer projection form, even though a Fischer projection allows for a lot more information about the monosaccharide to be given. I will never give you a Fischer projection and have you say, well, is this glucose or not? Only with respect to the cyclic ones. Now, because we're in biochemistry, this is much more complex. So you may be thinking like, oh my goodness, how am I going to possibly remember the structure of glucose? Well, I'm never going to have you draw glucose from memory, for instance, where you have a blank piece of paper and you have to draw it. Pick it out of a lineup with other ones? Perhaps so, and indeed so. And we'll start looking at that as we loop in a few other up to 5 representative monosaccharides, but again, recognized only as a Haworth projection. We have another one. No, first we'll notice back here on glucose. Glucose is a 6-carbon monosaccharide. We can count them. 1, 2, if we count them around, 1, 2, 3, 4, 5, 6. They're all 6-carbon monosaccharides. Another 6-carbon monosaccharide is known as galactose. Galactose is different than glucose, although if you just take a quick look at it, it would be difficult to tell. There is one structural difference. We'll talk about that as we move along. If you look at whether it's the Fischer projection of glucose and galactose or the cyclic Haworth projections, it becomes a slight structural difference becomes apparent. First of all, remember now, it does not matter the disposition of the oxygen on either galactose or glucose at the anomeric carbon. So here over on the right-hand side, here's my anomeric carbon, hydroxyl down, that's alpha. Anomeric carbon, hydroxyl up, that's beta. They see the same thing on the glucose, whether it's down or up, it doesn't matter because they're both glucose. Because remember, the alpha the alpha anomer can equilibrate into the beta. So they're both the same thing, really. But if you remember, if we look over again at the structure on the right-hand side here, any other hydroxyls in it are always specific. We never invert those stereocentries. It's always, we can invert around the anomeric carbon, down or up or down or up. The ones that we see within the ring that aren't at the anomeric carbon are always disposed the same way. Always. So that can help us when we look at structural differences between say, glucose and galactose. We never look to the anomeric carbon, the carbon furthest over to the right. But let's look at a couple of these, just to start getting used to structural differences. Let's look at the beta anomer of glucose and the beta anomer of galactose. So if you put those two together, you might think that they are very, very they look identical to me, and you'd be pretty correct because they look pretty identical. If we are careful, though, if we look over at the furthest carbon on the left in this Haworth projection, there is a slight structural difference. On that furthest carbon on the left, on glucose, we have hydroxyl pointed downward, down here. On galactose, we have the hydroxyl pointed upwards here. Remember, that one never changes because that is not at an anomeric center. That is always the case. So whenever we see a comparison between glucose and galactose, and again, I'll try to clean this up a little bit. We look over at the carbon. It's a six-membered ring with 6 carbons. We look over at the carbon on the left on both of them and ask, which way is that hydroxyl pointing? If it's pointing down, it's glucose. If it's pointing up, it's galactose. It's a very, very slight difference, but it is a difference nonetheless. And believe me, given some practice, you will be able to tell the difference between glucose and galactose. Remember, always, only I would give them to you in the cyclic form to tell the difference between. Let's look at the cyclization of another 6-carbon monosaccharide, but this time it cyclizes into not a 6-membered ring in a Haworth projection, but a 5-membered ring. Then this is a different type of Haworth projection, 6 or 5 being the only ones that we'll be showing you. And this would be with respect to the 6-carbon monosaccharide fructose. Fructose is one of our representative monosaccharides whose cyclic form I'm going to have you be able to tell the difference between. Now, fructose, again, if we count, we do indeed have 6 carbons right down the center in that Fisher projection. But in this case, while glucose and galactose were aldoses, meaning that their most oxidized carbon was the topmost carbon, in this case, fructose is actually a ketose, where that most oxidized carbon is 1 carbon shorter in the ring, 1 carbon down. It is not the top carbon anymore. So that carbonyl, remember, is right here, is where that cyclization happens with that C5 oxygen. So with that carbonyl being dropped by 1 carbon, the ring that is formed is dropped by 1 size, whereas before we had a 6-membered ring in glucose and galactose. Now the cyclic form is a 5-membered ring in fructose. Now notice, again, it's showing you this D-fructose. It is D. Well, it's always going to be D, so what we have here is just a bunch of extra stuff. It doesn't matter. But notice that in this case, here is the bond that we've just formed. Here is the bond we've just formed in this new type of Haworth projection, the 5-membered ring Haworth projection. In a 5-membered ring Haworth projection, we have, if we look at one of these, in this case it's the alpha anomer, we'll look at why that is in a moment, it is a 5-membered ring. And in this case, the ring oxygen is set sort of to the pinnacle, the very top, sort of the rooftop of a house, if you will. And that is in both cases. If you look at the structure down there, that ring is always going to be the case on a 5-membered Haworth projection being right at the top. As before, always that anomeric carbon is going to be over the furthest carbon over to the right on the molecule. And we then have just to look at the disposition of the hydroxyl on each of these. Let's clean this up a little bit. And we have the disposition of the hydroxyl on the molecule on the top here. That goes downward. That means it is an alpha anomer. The hydroxyl on the one on the bottom here points upwards. That means it is a beta anomer, again, just like we saw before. So that anomeric center can always either be alpha or beta. It doesn't matter. It's still fructose. Again, now let's count carbons on this, because it's very easy to think, well, I have a 5-carbon ring, or a 5-membered ring, thus it must be made of 5 carbons. It's not the case here. Let's count, and we're gonna follow this along. And this is actually, look, we've numbered these 1, 2, 3, 4, 5, 6 in the Fisher projection. We numbered them 1, 2, 3, 4, 5, 6 in the Haworth projection. Indeed, there is 6 carbons. This C6 flag up top is still waving in the breeze, but in this case on a Haworth projection is always on that the carbon furthest from the left. Just a structural difference. Not that I'm going to have you specifically being drawing these from scratch either. The reason, so what carbon is left over? We have a 5-membered ring here, but where's that 6th carbon? Well, here's the 5th, and there is the 6th right there. We have that sort of hanging off like extra baggage, baggage up here, this extra CH2OH. Similarly, on the beta anomer, we have that there. It's just the fact that if we look back over on the Fisher projection on the left, there it is. There's that carbonyl carbon is one carbon down on the chain, and therefore it becomes a 5-membered ring instead of a 6-membered ring. Now, as we'll see, fructose, being a 5-membered ring, is very easy to distinguish between the other 2 6-carbon monosaccharides. They were in a 6-membered ring. We'll put all these together at the end, and we'll slowly parse through them in order to tell the difference between them. But thus far, of our 5 representative monosaccharides that I'm going to be showing you, we've seen 3 of them. Again, I am only ever going to be asking you to distinguish between them from their structure in the cyclic form. So let's summarize our representative 6-carbon sugars up top here. There's going to be, so we have 3 of them. 3 of them, and these 3 have some similarity in that they all provide a nutritional role in biological systems. You think sugars, of course it's going to be a nutritional role. However, we're going to find a couple of other sugars or carbohydrates that don't play a nutritional role, but rather other roles as well. In any event, here we have our 3 monosaccharides, 6-carbon monosaccharides: glucose, galactose, and fructose. The first thing we'll always do when we start to having to parse between them and figure out which one is which is count the carbons. And in each case here, these are all 6-carbon sugars, or 6-carbon monosaccharides. Glucose, now, so we have some biological functions here, our nutritional roles here. If we look at that, if we look at glucose in particular, glucose, as I said before, is the primary energy source in animal systems. It's the go-to energy molecule for extracting energy for use for our own uses. The center monosaccharide, galactose, provides a nutritional role in that it is a structural component of milk sugar, the sugar that occurs in milk. It is not specifically milk sugar, but it's one of the structural components of that. Lastly, over on the right-hand side, our 5-membered ring, 6-carbon sugar, is fructose. Fructose, its biological role is a fruit sugar. So in many fruits, it's we tend to distinguish it as a very, very sweet type of sugar. That is why in many fruits that fructose is common in are very sweet, like strawberries, like bananas, certain apples, that sort of thing. It's a fruit sugar. All right, the final 2 of our 5 representative monosaccharides, the last 2 of them are actually 5-carbon sugars. So we can distinguish between the other 3 that we looked at, which were 6-carbon monosaccharides. These are 5-carbon. And indeed, as we move along, we can count those carbons. If we look at the structure on the left, for instance, I count 1, 2, 3, 4, 5 carbons. So that's the first thing we're going to be doing in these cases is counting carbons. To explain some of the salient features, these 5-carbon sugars, remember our 6-carbon sugars played a nutritional role. The 5-carbon sugars play a structural role. If we look, molecular structure, if you will, we have a structural role specifically in the scaffolding of genetic material, DNA and RNA. Now, the first sugar we're going to look at, or first monosaccharide, again, from the recognition standpoint, is always with respect to, is always going to be in its cyclic form, in its Haworth projection. The first one we see here is ribose. Ribose is the structural sugar in ribonucleic acid, or if you like the acronym for it, RNA. It's a genetic material we'll be looking at down the road. Over on the right-hand side, we have deoxyribose. Deoxyribose is the structural sugar in deoxyribonucleic acid, or DNA, maybe perhaps more familiar. Now, both of them have 5 carbons. 5 carbons to them, but they are somewhat structurally different, and it's really in the name that it's going to show the difference between them, if you will. If we look over on the left-hand side at ribose, and I'll bring to your attention this carbon right here. This carbon right here has a hydroxyl on it, happens to be pointed down. It's not the anomeric carbon, so this one will always be pointed down. Let's look at the same carbon on deoxyribose. That is right here over on the right-hand side. Notice there is no oxygen there. We have a hydrogen and a hydrogen. It is not connected to a hydroxyl. This is actually very rare in carbohydrate chemistry, because virtually always in carbohydrate chemistry, every carbon is at least connected to has one carbon-oxygen bond, almost always. Deoxyribose is an exception to that, a rare exception to that. So we're able to, if we have our 2 5-carbon sugars, the first thing we're going to go to is that carbon right over there. Is there an OH on it? If not, it is deoxyribose. If it does have an OH, it is indeed ribose. So we're able to use those structural difference to tell the difference between them. So now we've come full circle to the pinnacle, or the real question. Here are 5 representative monosaccharides. Go ahead and identify them all. And based upon all the structural features we saw before, we can indeed do that. The first thing to look at in these 5 monosaccharides is we have 2 of them are 6-membered rings, and 3 of them are 5-membered rings. But if we remember now that our 2 6-membered rings were either glucose or galactose, and those 2 were the hardest to tell apart. They had the most just hardly any structural features differences, but it is, so it's these two that are the 6-membered rings, not 6 carbon rings, 6-membered rings, which is a hexagon. So all we'd have to remember was, in this case, that our glucose had our C4 hydroxyl way over on this side downward, whereas galactose had that C4 hydroxyl upward. So we could immediately identify those two. And fructose would be the next one, because if you remember, it is a 5-membered ring with 6 carbons in it. 1, 2, 3, 4, 5, 6. It is the only 6-carbon monosaccharide as a 5-membered ring. So fructose would be that one. It looks very much like our other two structures, which are 5-membered rings, but these are the ones with 5 carbons. And if we remember, the 5-carbon ones were the ones that play the structural role. And so those two there was either ribose or deoxyribose, one or the other. Deoxyribose, so that would be the one without that oxygen, that very rare case at that carbon. So indeed, this must be the deoxyribose, and the last one would have to be, in this case, ribose. So we're able to, using these structural differences, work through them and be able to identify, if I gave you a test or a quiz or an exam, all 5 of these, you would be able to tell me which ones of them that they were. We finally completed the first component of this unit of study, that of the analysis of the monosaccharides, and now we're going to start to bring these monosaccharides together by covalently linking them into larger molecules of varying size. So the second type of manner in which we're going to be looking at carbohydrates is with respect to what are called disaccharides. Di means 2, so we're talking about 2 monosaccharides that are going to be covalently bonded together. And it is the nature of this covalent bond that derives a lot of the interest or salient features of disaccharides. So what we have to start are always, the way we're going to be viewing disaccharides, are 2 monosaccharides in the Fisher projection, the Haworth projection form. Always is the Haworth projection, where our anomeric carbons and oxygens are going to always be on the furthest carbon to the right, and we put them right next to each other. When we do this, we sort of push together the anomeric center of the monosaccharide on the left with the 4 carbon, in this case, of the monosaccharide on the right. And each of them happens to have a hydroxyl on there. So if we look at the nature of these hydroxyls, how we form a bond from there, ultimately we extrude water from it, and I'll show you right here. We'll look at HOH, that's water, and what is left, carbon, oxygen, carbon, is going to be ultimately connected together. So here we have 2 monosaccharides which are covalently bonded together into disaccharides by a specific type of bond, and that is known as a glycosidic bond. A glycosidic bond. This is the glycosidic bond that we see right here. So the glycosidic bond is also colloquially known as an oxygen bridge. An oxygen bridge, because here we have an oxygen, sort of this bridging gap between it. You can think of it as such, an oxygen bridge. So one couple of salient features regarding disaccharides. This is just one representative disaccharide that we can see here. And notice we got rid of water out of there, and there is water right there. What is left is this glycosidic bond. So now some salient features might be that, as I said before, our disaccharides are always going to have our monosaccharides expressed in that disaccharide in their Haworth projection form, always in that form. So, just clean this up a little bit here. So it is always assumed, as I said before, that these monosaccharides are of the D designation, so much so that we don't even need to say it. We don't even need to say it. So here now we have one representative monosaccharide, and we're going to find that it is contingent upon what the identities of the monosaccharides in each disaccharides are for the identity of the disaccharide itself. So we will ultimately look at that, look at a little more closely at the glycosidic bond, that glycosidic linkage, in the next slide. Let's take a closer look at a glycosidic bond in a disaccharide with respect to the anomeric centers in each of the monosaccharides which make up the disaccharide. So here is our glycosidic bond right here, our oxygen bridge. And what happens is when we have a glycosidic bond between two monosaccharides, we no longer have a hemiacetal at that anomeric center. Remember, a hemiacetal was COCOH, and that was very labile. It could open and close, open and close. But when we have when we have a glycosidic bond, we end up with a full acetal linkage, which is COCOC. If you'll remember, full acetals and full ketals had that diagnostic linkage. In fact, we can trace this out. We can find carbon, oxygen, carbon, oxygen, carbon in this. A full acetal linkage is not labile, like a hemiacetal linkage in a monosaccharide. It is locked. So this entire anomeric center is locked, and it will never open, it will never reopen until such a time as the glycosidic bond itself is broken. So let's play a little thought experiment with our representative disaccharide here. We're not really naming this disaccharide or looking at it too closely, just one representative one. And let's ask the question, what would happen if we submitted this disaccharide to a Benedict's test? Would it give a positive or negative Benedict's test? Now, if we'll remember, if a molecule or an unknown has an aldehyde functional group on it, it tests positive in a Benedict's test. If it does not have an aldehyde, it tests negative on a Benedict's test. Now, looking at this molecule right now, we see that there are no aldehydes on it. In fact, however, we see even more clearly that at this anomeric center right here on the monosaccharide on the left, that is locked. It is locked. It cannot open. And if it cannot open, it cannot bear its aldehyde, if you will. It cannot reform it. Because remember, this hemiacetal, COCOC, or this acetal rather, locks in that aldehyde. The hemiacetal, COCOH, can still open up. So while it might be tempting to say, if we treated this with a Benedict's test, it would give a negative Benedict's test. The reason being that we would give is because this anomeric center right here is locked, and it cannot open up to bear its aldehyde. However, what about this anomeric center over here? On the rightmost monosaccharide, that one is still labile. It is still a hemiacetal. So if the label hemiacetal now can still open up, and indeed it does. So while this anomeric center over here on the right now is closed, this can still open up back and forth. And look, here we now have an aldehyde, and that aldehyde will test positive for a Benedict's test. I guess the real rub here is that when we're looking at this the disaccharide over on the left-hand side, it is obvious there's no aldehyde there. However, we realize that we have a labile hemiacetal site here, which will open up to give to show its aldehyde, and thus being trapped and give a positive Benedict's test.